Hi everyone, myself Dr. Yogesh, Professor and HOD from the Department of Oral Pathology and Microbiology, Sri Rajiv Gandhi College of Dental Sciences, Bangalore. Today I am going to talk about amelogenesis, that is the formation of enamel, which is otherwise explained by the term life cycle of ameloblast. As we all know, enamel is the hardest structure in the human body. It is harder than the bone. So this enamel is formed by the cells known as ameloblast. So the cycle involved, the various stages involved are explained by the life cycle of ameloblasts. And this enamel formation begins in the late bell stage of the tooth development. This enamel which is formed is the highly mineralized tissue which requires mainly three factors. So the amelogenesis and the ameloblast should be taken care during its formation and development, which requires a good nutrition, the blood supply, the removal of the organic content and the various enzymes involved. So basically it is under the genetic control. This amelogenesis should be associated always with the dentinogenesis, because without the first layer of dentin formation, ameloblasts are never going to get stimulated. We call this effect as inductive effect. So one thing we have to remember here is the first tissue in the tooth which formed is the dentin, not the enamel. Only after a layer of dentin is laid down, the ameloblast will be stimulated, and which helps in the formation of enamel. Both enamel and dentin formation begins at the dentino-enamel junction. So this life cycle of ameloblast or the amelogenesis is explained by various stages and phases. We have the pre-secretory stage, secretory stage and post-secretory stage. And based on the function of the ameloblast, various stages are morphogenic stage, organizing stage, formative stage, maturative stage, protective stage and desmolytic stage. Okay, morphogenic, organizing, formative, maturative, protective and desmolytic stages. So this is the diagrammatic representation of the amelogenesis process. The inner enamel epithelial cells will be differentiating into the pre-ameloblast. So they change their morphological shape from cuboidal to low columnar, which later become into tall columnar cell. The various stages give rise to the various morphological appearances. There will be a development of a structure called Tome's process, an important structure which happens in the formative stage. During the maturation, once the full thickness of enamel is formed, the maturation happens by removal of organic content and the introduction of inorganic minerals. So once the full layer of enamel is laid down, these ameloblasts again become cuboidal in the shape. These are called post-functional ameloblasts because they have performed their function. What is the function? Formation of enamel. So we refer it as post-functional ameloblast, which covers the completely formed enamel till the tooth and the oral cavity. The first stage, pre-secretory stage, as I already explained, the odontoblast which differentiate from the dental papilla, lay down pre-dentin, the initial dentin matrix which stimulates the amelogenesis process. Thereby the inner enamel epithelium or internal enamel epithelial cells of the enamel organ differentiate into pre-ameloblasts. This is the diagrammatic representation showing how the inner enamel epithelial cells are differentiating into pre-ameloblast and ameloblast. So this ameloblast, once they are differentiated from the inner enamel epithelial cells, as you can see, a first layer of dentin is formed. This dentin, once it is formed, the basal lamina or the basement membrane which is separating the inner enamel epithelium from the dental papilla will be disrupted. So the shape of the ameloblast become tall columnar. There will be reverse polarity of the nuclei. The nucleus which was in the center pushes away from the basement membrane, that is towards the proximal surface or the proximal end, we call it as. The one towards the secreting enamel or dental papilla we call distal end. The one towards the stellate reticulum area is called proximal end. So once the odontoblasts are differentiated, the basal lamina disintegrates. Meantime, the ameloblast secretes formation of the enamel. The initial enamel which is formed is the aprismatic enamel. So the length which has been pre-ameloblast length and diameter is 40 micrometer and 2 to 4 micrometer. So the next stage is the secretory stage. This secretory stage corresponds to formative stage of the amelogenesis, life cycle of the amelogenesis, wherein there will be development of Tomes process at the distal end. So these are the cone shaped projections from the distal end which jet into the forming enamel matrix. So this forming enamel matrix, thereby if we observe under the electron microscope, which gives pit and the fish scale. So this is the picture how the distal end of the ameloblasts are jet into the forming enamel matrix. The Tomes process are delineated by a process or a structure called terminal bars. So which we will talk about later in the maturation stage and describe the two types of terminal bars, that is the proximal terminal bar and distal terminal bar. So this is what I was talking about, the pit formation, where the Tomes process jet into the forming enamel matrix. So these Tomes process gives rise to an appearance called picket fence appearance, or we call it as sometimes tombstone appearance. Okay, picket fence. We can see how they jet into the forming enamel matrix. As the enamel matrix is laid down, this Tomes process, they retrieve towards the stellate reticulum area. This is the mineralization on the forming front of the enamel. So this is how typically we can see a picket fence or a sawtooth appearance, how a sawtooth which used to cut the trees. So sawtooth pattern can be seen in the electron microscope. As the enamel matrix is laid down, they retrieve towards the stellate reticulum area or the stratum intermedium area. So once the ameloblasts are developing the Tomes process, meantime matrix vesicles are happening. They help in the mineralization process. The mineralization of enamel is very important because this doesn't have a lag time. The matrix deposition and the mineralization happen simultaneously. So once the enamel matrix is laid down, the ameloblast will be retrieving towards the forming stratum intermedium area. Meantime, we can see the dentin formation by the odontoblast. Both happen simultaneously. So we can see something like a mirror image of the two cells. We call it as they look like a mirror image. Ameloblast on the one side and the odontoblast on the other side. So as I said, both the secretion and the mineralization happen simultaneously. So there is no undistinct zone of enamel, unmineralized zone of enamel as we see in predentin, which is the unmineralized hypocalcified or the less calcified dentin zone which is seen during the dentinogenesis. So we can see the matrix vesicles within the ameloblasts. So once the full thickness of matrix is formed, the secretory phase ends. As I said, the terms process retract, the distal end becomes flat. So there will be an aprismatic enamel on the surface of the formed enamel. So thereby the 4 ameloblasts contribute to a single enamel rod or single prism. Each ameloblast help thereby in the 4 prism development. So each prism elongate incrementally. The daily increment we call it as cross striation, and the weekly increment results in the enamel striae, we call it as striae of Retzius or the incremental line of Retzius. So this is what I was talking, how the pit in the interrod region, the pit happens because of the Tomes process. This is a low power view, and this is the high power view of the electron. Next come to the transition stage, which corresponds to maturative phase of the life cycle, maturative and the protective. This maturation stage is very important, wherein the fully formed enamel, initially it is more porous, which has high content of water and protein and low content of mineral. So which has to be replaced by the highest calcified structure in the body, that is the enamel has 96% inorganic. 96% of calcium, phosphate, hydroxyapatite crystals and other minerals like fluoride, magnesium, manganese, and only 4% of organic content. So this enamel maturation happens by the removal of various enamel proteins. So we will talk about enamel proteins. They are the ubiquitous in nature. They are different from the proteins in other part of the body. So maturation, 30% enamel will be come down to 1%. So what are these enamel proteins? So there are 2 types, mature and immature forms. In the immature, 90% to 95% are amelogenins, and to certain extent 5 to 10% is non-amelogenin, which is otherwise called as enamelins. Within this enamelin, we have the ameloblastin, tuftelin, sheathlin, proteinases. So all these enamel proteins helps in the during the maturation stage. They wash away, they will be removed by the distal end of the ameloblast. So amelogenin, which is the major contribution, they are hydrophobic in nature. They tend to clump or aggregate. They spread throughout the whole developing enamel thickness. They are helping significant in the production of the large hydroxyapatite. Enamelin is located in the prism core. They control the growth of crystals by an inhibitory action, and they are one of the largest enamel protein. Ameloblastin, which is otherwise called as amelin or sheathlin, which is initially located in the prism core. The main function is generation of the prism structure. Also it is produced by the Hartwig's epithelial root sheath during the root formation. Tuftelin, which is another enamel protein to minor extent. This tuftelin is in one of the signaling molecule during the epithelial mesenchymal interaction, mainly located in the dentino-enamel junction, and it acts as a nucleator at the commencement of initial enamel mineralization. Amelotin is another protein which is secreted mainly during the maturative stage of the amelogenesis. So this is expressed later than the other enamel proteins, mainly associated with dental lamina, the basal lamina or the basement membrane. The main role noted is helps in the cell adhesion. So these are the various enamel proteins and their functions. So in the maturation stage, most of the formed enamel, which has 65% water, 20% organic and only 15% inorganic, has to transform into 96% inorganic and 4% organic. So how it happens? By the growth of the crystals, the increase in their width and their thickness, subsequent reduction in the intercrystalline space, and meantime ameloblasts move calcium and phosphate and carbonate into the secreted matrix. So these are the changes which are happening mainly during the maturation stage. The distal end of the ameloblast shows the morphological changes. Microvilli, or we call it as brush border or ruffled border appearance, which helps in the introduction of inorganic crystals into the formed enamel matrix, and the distal terminal bar are very tight here and the proximal terminal bars are leaky. When we come to the smooth ended ameloblast, we can see here the distal terminal bars are leaky or open and the proximal terminal bars are tight. And this smooth ended ameloblast helps in the removal of the water, organic material and enamel proteins. So thereby the maturation happens. So as I said, the enamel protein content reduced from 30% to 1% during the maturation stage. Ameloblasts remove this water and degrade the enamel matrix protein. The crystals increase and expand in their thickness. So how this pattern of mineralization happens? It mainly begins from the cusp tips and it extends or progresses towards the cervical region. So cusp tip is the first site, it extends towards the cervical or cemento-enamel junction. This is the pattern of mineralization during maturation. So once the maturation happens, we enter the post-maturation stage. Here we see desmolytic stage and protective and desmolytic stage mainly. The ameloblasts, which are tall columnar, will become flattened to cuboidal shape. So a thin layer of amorphous basal lamina which covers the unerupted tooth, which is called enamel cuticle, primary enamel cuticle. So this will be lost once the tooth erupts into the oral cavity. So the reduced enamel epithelium is the term, as we know, which happens because of the post-functional ameloblast and other cells of the enamel organ like stellate reticulum, stratum intermedium, all together forms the reduced enamel epithelium. This reduced enamel epithelium covers the unerupted enamel always till the tooth erupts into the oral cavity. Once the tooth erupts into the oral cavity, this reduced enamel epithelium joins with the oral epithelium to form junctional epithelium or attachment epithelium. This is the picture. Again I am showing how the morphological changes. Initially the inner enamel epithelial cells, which were low columnar to cuboidal shape, become into the pre-ameloblast, which are columnar in shape. There will be reverse polarity of the nuclei. Nuclei goes from the center to the proximal end, away from the basement membrane. There will be development of the Tomes process which extends from distal terminal bar. So these terminal bars attaches one ameloblast to the other ameloblast. So there are two types, proximal terminal bars and distal terminal bars. They play a very important role during the maturation by opening and closing at the proximal and distal terminal bar region. So once the ameloblast completely matures, the ameloblasts are no more function. So we call it as post-functional ameloblasts. So this amelogenesis entirely is controlled by various genetical hormones, enzymes, and a good nutrition is enough. Without a good nutrition, the ameloblast cannot lay down a good quality of enamel. So desmolytic stage, what happens? That reduced enamel epithelium secretes various enzymes. These various enzymes degrade the overlying collagen. Once the overlying collagen is degraded, which helps in the eruption of the tooth. So another function of reduced enamel epithelium is it protects. How? But that's why we have protective stage. It protects the deposition of cementum on the enamel. Otherwise, if there is no reduced enamel epithelium, cementum from the dental sac would have been formed on the enamel surface. So various clinical consideration, as I said, a good nutrition various hereditary factor and environmental factors play a role. If there is any deficiency or a genetic mutation, any trauma, injury, and the other environmental factors like systemic diseases, syphilis, scarlet fever, measles, all can damage the ameloblast, which results in amelogenesis imperfecta. This is basically an hereditary form which have various types, hypomaturative type and hypoplastic type. In the environmental, again, if there is any damage to the deciduous tooth, which can in turn damage the deciduous tooth bud can damage the permanent tooth bud. So all results in hypoplastic. Thank you.